So I'm very glad that we have the first speaker here, Aleksandra Kolodziejczyk, who was
Hola.
Aleksandra Kolodziejczyk, sorry, Ola, who is actually one of the first to analyze single cell data. So please explain us what it is all about.
Hi, welcome everyone. So as Eva said, I work in the single cell field for a while now. So I started in 2012 in my PhD. And what I would like to show you today is some of the methods that we use since then, and about kind of options that you have for the experimental part, how to make the data. And I think this is very important, even though some of you will never do experiments, to understand what kind of data you have, from which technology it comes from, and what kind of biases, what kind of information it carries. So, so you know what to expect, and I will show you some examples why it's nice to know how your data was made. I don't have many slides. It's all pretty simple. So if anyone has any questions, you can interrupt me. There's no problem. We have a lot of time today. So single-cell RNA sequencing, or single-cell sequencing in general, was a method of the year in Nature Methods in 2013, so already a while ago. So it was recognized to be something very useful very early on, because the first paper about single-cell RNA-seq was in 2009 from Azim Surani's lab. And then in 2013, there were maybe like 7 papers or something like that. So, it was recognized to be important very quickly, and it really boomed within the last couple of years. So why do we do it? And I showed this slide for the last 5 years, really the same slide. So basically, I worked in my PhD on stem cells. And stem cells in culture are not the same, all of them. So they are heterogeneous. And there are 2 genes that on protein level, you have OCT4 that is expressed more or less the same in all the cells. But there's this other gene called NANONG, also very important transcription factor for embryonic stem cells. And you see some of them express it quite highly, others quite lowly, in quite low amount. So to study problems like this, in past people use FACS, or you could use microscopy. But obviously this has many limitations, especially the big limitation is that you have to pick the genes that you're going to study. And now when we do all these big experiments with lots of single-cell RNA-seq data, we see that actually people were pretty remarkably good in picking the genes doing FACS, because they really could see actually almost all populations. I'm really shocked actually. Sometimes. So to study these more heterogeneous populations, single-cell RNA-seq is interesting. So in the respect of cancer, obviously, it's a very heterogeneous situation where you have cells coming from different clones. So both on RNA level and on the DNA level, they are heterogeneous. So using these kind of methods is very useful. And yeah, it's another slide that I show for the last 5 years. When you have this kind of mixture of different cells and you do bulk RNA sequencing, what you get an average of all of these cells. And I saw someone comparing this to making a smoothie. You can either have like a fruit salad where you can identify different fruits that were there, apple and peach and so on, or you can make a smoothie. You probably can feel the flavor of all the fruits, but if there was one grape there in like a big bowl of a smoothie, you probably will not be able to feel it, even though it was there. So it's something like that. You have this average that doesn't really represent any of the components, and on top of that, there may be some things that you are not able to see. But obviously, with the single-cell methods, you can see the gene expression in every cell. You can identify cell types, you can see the frequency of each cell type, so you can see which cells are more abundant, which ones are less abundant, so that's also very interesting, because when you compare between different experimental models, between different conditions, you can see this changing. And this is an unbiased approach. I think it's also very important to note that we see, well, we don't see all the genes, but we see all the highly expressed genes, which which allows us to do it in an unbiased way. So most of this talk will be about how to do the single-cell RNA sequencing. I'll go slowly, step by step, through all different methods and all different steps that you have to do to get the data, and finally you can actually analyze it. But I wanted to show these figures, which is really nice, of my colleague Valentin, who plotted a number of cells in study against the publication date. And one thing you can see, the number of cells that we get is really increasing dramatically over the last couple of years. And also he pointed here some of the more methods that are more often used. So the first paper that we were doing with John Marioni was here with the fluid IMC1, so we were the first to use that technology. That's now basically obsolete. No one is using it anymore. Yeah, you can see it's increasing. So I will talk about all of these different methods like SmartSeq2, MARSeq, CellSeq, 10x genomics, which is something that many people use at the moment. And I will show you what kind of advantages and disadvantages are of these methods, and I'll explain you how do they differ, what's similar between them. But basically, the general workflow of single-cell RNA sequencing experiment looks like that. So you have a tissue, usually it's a solid tissue, like a liver, like a tumor, like anything. First, what you need to do, you need to dissociate the tissue, and this is already not trivial. So then you get single-cell suspension, so each cell is not linked to other cells by matrix. Then you need to isolate single cells, and this sounds also very simple, but again, it's not trivial. Then you need to lyse the cells and get the RNA out. Then you need to reverse transcribe it to get cDNA. But obviously, it's single cells, it's not so much of the material there. So it wouldn't be enough to sequence. So we need to first amplify it a little bit, either with PCR or with reverse transcription, with IVT, sorry. And then with reverse transcription, so it's like a different way of amplification. Then we get more, and then only we can do a library to sequence it. And most people use Illumina, but now I think more and more people are interested in long reads, like with PacBio or Nanopore as well.
Sorry, what's IVT?
As in vitro transcription. Right, so you basically go from cDNA to RNA, and then you go back to DNA. But I will talk about this deeply, more deep. This is just a workflow, so you can see more or less what happens afterward. So the first thing is tissue dissociation. So obviously it's like a chunk of something, like piece of chicken or some, it's a big, big thing. So what you need to do, you need to digest it, and there are different ways. So you can either do it in an enzymatic way or mechanically, or combine this method. And this really depends on the tissue you work with. So for example, if you work with spleen, usually you do mechanic dissociations because cells, it's enough to smash it and then cells already are single cells. But if you work with, for example, liver, I work a bit with the liver lately, so there you need to do enzymatic digestion. And then the enzymatic digestion can be done in situ or in vitro. So either you digest the organ already inside the animal or inside the... So also you can do it with I saw recently on Protocols.io, a very nice digestion of human liver outside the human, but with perfusing the system with enzymes. Very, very nice. So there are different options here. And then the mechanic dissociations, different types of smashing, cutting, and so on. And you can combine these. So that's often what people do. They cut something in small pieces and then they add enzymes like collagenase, proteases, and so on. There are some commercially available things like GentleMax from Milteney. So you can also use these. They usually work pretty well. And something I wanted to show is this paper from Alexander van Oudenaarde, who last year shown, and something that many of us saw already before, that this digestion may lead to changes in gene expression in the tissue. And interestingly, the pattern of gene expression changes that comes from the digestion seems to be similar between the tissues. So he here shows examples of some genes like FOS, like June and so on, that tend to be upregulated if the digestion is done too long in 37. If you keep your tissue for 2 hours in 37 digesting with collagenase, it's very likely that these genes will pop up. So when you analyze your data and you see some subpopulation having these genes, be careful, because this may be not a real signal, but may come from digestion. So this is something to keep in mind.
So the samples he analyzed, so the cells that he analyzed, did they come from normal tissue or also cancer tissues?
These are normal tissues, but he used it from different tissues, very different tissues. We see it in
cancer, it would be the same, but it would also react in the similar manner.
I don't know. I never worked with cancer. I don't have this, but it's very likely that yes, because this seems to be. I know that in liver, in brain, in I think also in kidney, this pattern occurs. So these are very different tissues. Okay.
Thanks.
Yes.
Work only on the fresh tissue, or can it be used also on like frozen or fixed samples?
Yes. So you can use frozen samples. It's you need to freeze it in the right way. The quality usually drops. There are some protocols available. Again, you would have to find appropriate protocol for your particular tissue, but it is possible. Yes, but the data is worse than the data from fresh tissues in general. So I would suggest, if you can, to work with fresh tissues. Definitely. So that's the first thing you do. The second thing you have to do, you need to capture single cells. And again, it may seem easy, but it's one of the trickier parts and one of the stages where most technology development is happening. So in the first paper from Asim Surani's lab in 2009, what they did is they just manually picked under the microscope. They had 5 oocytes in that paper, and basically they just picked them. But obviously now we want to sequence thousands of cells or hundreds of thousands of cells. So this is no longer an option. Meanwhile, people started sorting single cells to wells and then doing protocols in single cells, either 96 well plates or 384 well plates. So then this is like additional option that you have. This obviously increases the throughput, but has some limitations. You can do hundreds, you can maybe do a couple thousand, but not hundreds of thousand cells with this kind of approach. Then there is this Fluidigm machine that we also used, which is like a chip which allows capturing single cells. Many institutes have these, but I don't think anyone is using them anymore because these chips, they only catch 96 cells and they are pretty expensive. So other technologies basically took over this. Then in 2015 we had these 2 big papers about droplets, and this was a big revolution in single cell field. So basically what you can do is you can capture your single cells in these aqueous droplets in oil, and then each of these droplets can be like your small bioreactor where you perform all these steps of reverse transcription and so on. And these droplets allow us to do many of these cells at the same time. And at the moment we have this technology called 10x genomics. Everyone has probably heard about it. So you can just buy a box and a kit, and it's very simple to start this way. Even if you don't have experience with single cell RNA-seq before, it's really pretty easy to start. Then there's another option that you can do is you can do pico wells. So it's basically like well plates but with tiny wells. So that also increases the throughput. And then recently, people started to do so-called in situ barcoding. So basically what this does, you permeabilize the cells and you do stepwise ligations of barcodes. And you do it in a combinatorial way. So basically you have your sample, you split it, let's say in 10 samples, then you ligate 10 different barcodes. Then you put them together, you separate them, then you ligate again, and do it a couple times. So each cell this way will have different barcode because the combinations are so many. So this is something that's coming up at the moment. So once you capture your cells, and once they are separated.
But then you're, sorry, in this barcoding, they're
Mhm.
still together, right? So
Yes, they are still together. Yes, but
so when you then, so you don't separate them? First?
no, but basically you add the barcode. That's enough.
And then you sequence also the barcode.
Yes.
And then you know which cell has been.
Yes, exactly.
Okay.
Exactly. So the first thing then you have to do to get your libraries is you need to do reverse transcription. And there are two main things that are important here. First is in cells we have mostly, the RNA is mostly ribosomal RNA. So at this stage we want to get rid of ribosomal RNA or not to proceed with it to the next step. And the second thing is we want to get double-stranded DNA. So reverse transcription in general can be done in two different ways. You can do oligotype priming or you can do random priming. And in general, in single cell experiments, we mostly do oligopriming and mostly with an anchored primer. So basically it has, it doesn't prime wherever in the poly A tail, it just primes at the end of it. So we don't do reverse transcription of the tail. There was a paper sometime around 2014 which used random primers which were not so random, meaning that they were depleted of sequences that would align to ribosomal RNA, and this way they were amplifying only messenger RNA, but it didn't work very well. No one really picked up that method. So it's usually, it's basically always priming from the poly A. And this way we do reverse transcription only of mRNAs because ribosomal RNAs don't have the tail. And then there are two ways of getting the double-stranded DNA. So one thing is we can do homopolymer tailing, where after the reverse transcription on one side, then you do tailing of A's, and then you go backwards. Or you can do template switching, which many people also do. For example, SmartSeq protocols, it's template switching protocols, where basically the template switching happens as well in here. There's tailing of C's, and then there's a special primer called template switching oligo that allows to go backwards, and this can be done in one reaction. So after this reverse transcription, you get to your RNA, your cDNA, and then you need to amplify it. And there are two major ways to amplify it. You can either do PCR or you can do IVT. And PCR is mostly used. Most protocols, except CellSeq and MarSeq, use PCR. Because it's a bit simpler to do and faster. But PCR is not a linear amplification method, so some people don't like that. So basically once you have your cDNA, then because you have either these T's and A's here, on the other side you can use this as an anchor to PCR, or here you have the C's and G's that happen from the template switching reaction. So you can also use them as an anchor to PCR. With in vitro transcription, thing is a little bit different. So you, once you have the cDNA, when you do a reverse transcription in your primer, in your poly T primer, what you have a promoter from which the IVT can happen. So usually like T7 promoter. And then you go from the cDNA again back to RNA. This is a linear reaction. And then from this RNA, you go back to cDNA. There's a disadvantage of this is that in general, reverse transcription is not a very efficient reaction. So not all of the molecules get reverse transcribed. Yes?
But there are protocols also for microRNA, right?
Yes, they are.
So
But they're
what are they based on?
But they are usually targeted.
Sorry?
They are usually targeted. So basically, you have a library of microRNAs that you target, and then you amplify those targeted. At least those that I know. But now there are so many things that there may be something that I don't know. So you get this amplified RNA, and then you reverse transcribe it back to cDNA. So basically, there are twice reverse transcription, and that usually leads to a little bit lowering of the complexity of the library. But then it's linear, so there are two things that you can look at here.
So when you say linear, what do you mean by this?
So it means that in PCR, from one, the amplification is like exponential, and here the amplification is linear. So it's just amplifying in a different way. Yeah. But
Exponential sounds much more efficient, right? So when you say that some people don't like this,
it's because then the things that are highly abundant get overrepresented. And this is something that you don't want.
So the linear keeps the actual...
Yes, it's better in keeping actual abundances in the same way that they really are.
And that's important for gene expression, right?
Yes. But we have this new thing that's called UMIs that solves the problem. At some point, we realized that there's this issue of over-amplifying things because you have to do two PCRs, one which is this amplification PCR, and there's another PCR to make the library. So there's lots of amplification. Some molecules get overrepresented, and we don't really know whether this abundance comes from the PCR or whether it's real that something is expressed to some level. So we started using these unique molecular identifiers that basically are barcodes that are added at the reverse transcription stage. So then what we do is we go through the whole protocol. We check. So we have two. I have this example actually here. We have two cells. We do reverse transcription where we incorporate this UMI. So each molecule gets a different barcode. Then we do all these amplification steps, and we end up with some data on our computers. And what you see is that we have this molecule, even though it's 4 times, we read it as 1. Because the barcode it's the barcode that we care about. It's not the number of times something occurs. So we start counting barcodes rather than counting the reads. So because we know that each barcode comes only from one RNA. And this way we can correct for all these amplification biases that are occurring during the library preparation. So it's something that I strongly encourage, and I think at the moment everyone is doing it. Except it's a bit difficult if you do full-length methods like SmartSig. So that's something I'm going to talk now about.
But is there a chance that some barcode molecule will not ever be amplified?
Yes. Yeah, of course, most of things are not there, yes. Yeah. So there were some estimations.
Where does this come from? So at which step does it occur that you missed these?
So basically what happens is the first step, the reverse transcription, is pretty inefficient, and we know that we lose there, depending on the method, 80-90%. These are the estimations that were done. So then we are left with 10-20% of the RNAs only, and then we lose more a bit later as well. But most losses are done at the reverse transcription stage. So then when we see, so basically that's actually important when you analyze the data, is that when you don't see something, it doesn't mean it's not there. You can only know the things that are there. So it's important when you analyze to realize that.
I'm sorry, do you always close the same?
It depends on the method.
I mean, for example, the same genes, you always lose the same genes? Or
No. It's well, I'm not sure if we know actually if we always use the same genes, but it's assumed to be like this. No, yes, random. Basically, it would affect the same genes in the same way.
Or maybe it depends on the length of the transcript?
No, we don't think so. It may depend a little bit on the length of poly-A tail. If you don't use the anchored primer, then maybe. But no one really measured these things, at least not that I know of. Yes.
So when you say that we lose 80 to 90% of
Yes.
Do you say it's coming from the messenger RNA that we lose, or is it the other RNAs that we lose in the process?
No, it comes like basically these genes are not reverse transcribed, and or they may be lost a little bit also afterwards in the next steps. But mostly it's reverse transcription. Yeah. So once you have your cDNA and it's amplified, we need to do libraries, and there are basically two ways. It's also important for analysis later. You can, for methods where you PCR the whole, when you amplify in PCR the whole transcript, you can use Nextera, or you can do homebrew transposase. Do you know how the transposition in Nextera works? No? Okay. So basically what it is, it's actually pretty ingenious. It's just a kit from Illumina. So what you have the DNA. It can be done on DNA, it can be done on RNA, like cDNA, doesn't matter. And what you have is you have these transposases that cut the DNA. And while they cut, they also introduce little overhangs to the DNA. And this way, you end up with a fragment that has these overhangs added. And then to these, you can do PCR, and you can introduce barcodes, because you need more barcodes to, if each cell is in a different pool, in a different, like well, for example, then you need barcodes so you can pull everything together to sequence. So these are basically barcodes just to see the different samples for the sequencing. And so you do the PCR, then you get your fragment, and this fragment can be sequenced. While there are other methods where and this way you can actually sequence the whole length of the transcript. So if you're interested in splicing, or I'm pretty sure Peter will talk about Velocyto, which is really cool. And there also, this is useful. It's a method that they developed with Stanley Nurse. It's amazing. So for some methods, you want to see introns, or you want to see the full length for splicing. So then this kind of library prep is useful. But most people, what they actually want from the single-cell RNA sequencing is not the full length. They just want to know gene expression of each gene. And then it's cheaper just to sequence the very end, either on 3' or on the 5'. Because if you don't sequence, let's say your gene has 2 kb and you sequence only 100 base pairs from the end, it's way cheaper than sequencing the whole thing. So most people at the moment use methods that take either 5' or a 3', and they basically you would fragment the DNA and select for the end, and there are different ways to do it. But what is important is that you end up with a fragment that's just the very 3' or a very 5' end, in comparison to the methods where you have coverage throughout the whole sequence. And I just wanted to show you, because last year there were 2 papers that shown some comparisons between the methods, and especially comparisons in respect of number of detected genes and sensitivity of these methods. So this is one paper where they actually have done with the same cell type, basically the same sample. They sequence it with 6 different methods. So what they've used is they used CellSeq and SmartSeq on C1, so on this Fluidigm machine. They used a DropSeq, so a droplet-based method. They used MarSeq, so this method that was developed in Ida Amit lab in Weizmann, where I work now. They use this ScriptSeq, which I think is the method of this particular lab, and they use SmartSeq too, which also is quite commonly used method. It's quite nice. And you see these methods are very different, so they are different in respect of capture. They are different also here in a respect that some of them are full length, some of them have UMIs, some of them don't. So, and then they've done this experiment. They sequence all the cells coming from these different methods, and they have looked at sequencing depth and the number of genes that you can detect. So from this, we see that the highest number of genes that we can detect is with SMART-Seq2, comparison to the other methods that they have done. But we can also see that these other methods, so Script SMART-Seq2 on C1 and CellSeq on C1, are also performing quite well. MARSeq is quite worse, and DropSeq as well. But DropSeq has a big advantage of having really many cells. So basically, it's kind of a trade-off between having a little bit less genes detected, but having quite a lot of cells.
What's the scale? I'm sorry, because we can't see. What's the difference in number of...
So this is here, you see 2,000, 4,000, 6,000, 8,000.
So the number of cells, how does it enter this analysis? It doesn't matter, or the depth is over all cells together?
This is just basically, yeah. I mean, the average number of genes detected at this depth.
Detected means in at least one cell.
Yes. And here is the other method that my colleague Valentin have done, and it was done a little bit differently. So he took different data from different papers, and many people, when they do the experiments, especially in the beginning or when they want to validate the quality of their new method, what they do is they add this spike in called ERCC. These are 92 molecules of RNA, polyadenylated RNA, that you can add to your reaction to control for all the technical aspects. You can, because you know what you are adding and you know in which amount. So knowing this, you can see detection limit in molecules. So you know that you put there, let's say, 10 molecules. Well, do you detect this 10 one out of these 10 or not? So basically, this is this detection limit he calculated from the ERCCs from these different experiments. And again, what you see here is that methods that are best are the methods on C1, CellSeq, in this case, CellSeq, StartSeq, and SMARTSeq, all on C1 are the best. Then, I don't actually know, this is not a very common method to this BAT-seq, but then there are all the InDrop, Chromium, and DropSeq. So these are three methods, droplet-based methods. So they actually perform quite well. And then there are other methods, like in this case, SMART-Seq2 is higher, MARSeq is very high. For example, GNT-seq is a combined method where you do genome and transcriptome, and it's quite low because you need to physically separate RNA from DNA, and the losses at this stage are quite high. So this kind of, so you can always go back to these papers and the previous paper as well, and look at this parameters to choose the method that you want to use, or to understand how this method the data you got from somewhere else compares to the others. So I really recommend these two.
What's this parameter, M or number of M?
Actually, I don't know. I don't remember. Sorry.
Number of cells, maybe.
No, it's not number of cells, for sure not. No, because like in these methods you have thousands always, right? So it has to be something else. Maybe it's just some parameter here calculates of like which one is the best or something like that. And something that I usually get questions about is the cost. So I took from one of the papers, some people estimated cost. Obviously the costs are really dependent on where you work, because in some places sequencing is way more expensive than in other places, and sequencing is really big cost of this. So I was recently shocked that Sangha paid like half of the price for sequencing that we pay at Weizmann. So it can be a really big difference. But what you can see is that that the cost per library, so per one cell, can differ from like $1 or even less than $1 to even 30. So really, depending on the method you use, the costs vary a lot. So sometimes having a little bit lower quality, but you can have something way cheaper, so you can do more cells. So that's something to pay attention. And also, the minimal cost is important, because in some of these methods, you need to buy a whole kit, you need to do some number of cells at the same time, and that drives the cost higher again. And for example, in SmartSeq2, the expensive part is the transposase, the kit from Illumina. But if you can make the transposase at home, so you can express the protein and make it in your proteomics facility, which is not trivial because many people failed. So then you can drive costs down 10 times. So that's something important. I just wanted to mention that there are these combined methods where you can combine analysis of RNA sequencing with other genomics from a single cell. And there are different ways to split the cell. So you can either pull the RNAs using beads that bind to poly-A tail, or you can kind of lyse it gently where you release the cytoplasm and separate it from the nucleus. And then you can analyze the nuclear DNA separately from the RNA. And then basically on top of the RNA analysis, you can have DNA, analysis of the DNA, and then this can be either just simply genomic sequencing, or you can do also analysis of methylation, for example, which was done also when we were in Sanger, which is nice. I also not talk much about it because I saw that there are talks about it as well. So there's also big trend now about spatial transcriptomics, which is also very interesting because when we do this RNA-seq in a standard way, because we digest the tissue, we don't know which cells come from which region, and especially, for example, in cancer, it probably would be very interesting to see different regions of the tumor. How do they look? And which things are next to each other. So for this spatial transcriptomics methods would be very interesting. And basically you can divide them on this single molecule fish based methods. And there are different kind of versions of it. And then there are things based on sequencing in situ. And then there are these tissue reference map based methods where basically you still digest your tissues, but then you kind of computationally trace it back to some, because you have some data from imaging. It can be even very simple data from imaging. So this is something that is also very interesting lately. And I also have a couple things to talk about what kind of questions can be answered with the single-cell RNA sequencing. I have some simple examples. So these are some major problems and major questions that people do at the moment. So studying the heterogeneity, characterizing the population, looking at the new cell types, looking at the transitions between cell types and cell states, inferring gene regulatory networks now, also looking at the cell-cell signaling, and some looking at the transcription kinetics as well. So one example where single-cell RNA-seq actually started is looking at the early embryo. I think basically it started because these guys wanted to sequence oocytes, and they just had very few of them, so they had to develop some method to do it. So early with this kind of early embryo studies, basically that's the only way. Otherwise, the standard methods basically don't work here. And this is really a nice example of study of the early embryo, where you can see different stages of the embryo development. You can see how nice trajectory they have. You can study gene expression changes throughout this process. So then you can also, or like most of the studies recently, what do? Is they characterize cells of cells in particular tissue, often in health and in disease, to compare to see what kind of differences occur. Here's the example of the human pancreas. You can see all the different cell types and the different gene expression in these cell types. And then you can even zoom further, and then within some cell type, you can look at the heterogeneity. So you see then within these beta cells, they observe heterogeneity of unfolded protein response, or in the ductal cells, they can see some heterogeneity that comes from different positioning within the duct. So these are kind of questions that many people at the moment do. Then a very important part of single-cell sequencing is looking at the transitions between states. So this is often in development or differentiation, also in responses to stimuli, like when you add some molecule, like for example LPS to macrophages, they start to get activated. So you can follow this process, see how temporarily things are changing. And also the transition between cell states is cell cycle. So you can actually identify in which cell cycle stage your cells are. You can look at the cycling behavior. We were even able to kind of use this to the heterogeneity and differences between cells to kind of estimate which cells cycle faster. So, so these kind of things can be done as well. You can also look at the gene expression modules, because if you have a perturbed system, you can see genes that correlate with each other are usually within the same gene regulatory network. So you can identify these networks. Often you find genes that you already know in the network, but then you can find new players that are involved in particular networks. So especially interesting some transcription factors that are there. And I wanted to finish to thank, especially because all my single-cell work I did with Sarah Teichmann and with John, who's almost like a supervisor to me as well. At the moment, I'm in Eran Elinav group at the Weizmann Institute, and we study microbiome and the single cells as well. And these are my funding, so EMBO and Marie Curie. Thank you. If you have any questions. No questions.
Maybe I can ask a question. So you said you didn't work on cancer yourself.
No.
Is this? Do you have any idea how this is then applied in terms of cancer? Because we also have different types of cells, right? So, did you? Are you familiar with this type of analysis that's done in cancer?
So there are plenty of papers studying cancer. I think like in Aviv Regev's group, there were several. Ido Amit is now also working on cancer, like on lymphoma. So there are many. So some things that people study is so they are basic two sides of the story. One side would be looking at the cancer cells itself, looking at the DNA and so on, but that's kind of maybe less of what I'm interested in. But the other thing is to study all the cells around the cancer, so the immune system, and to compare it to the healthy state. And there's like growing a number of papers looking at that, so I'm pretty sure you can have a look at this.
Okay, so thank you very much.
I don't know how much you know about it, but you know, the identification of gene regulatory networks, how much better do you do with single cells? I mean, it should be much better, right? Not having the average, but the single cells.
So myself, actually, I've never done anything in not single cells, because that's true. So I don't know, actually. Personally, I've never compared. But it works. Like in my hands, we could identify like new transcription factors that are in the pluripotency core network. So yeah, I encourage people to try. I mean, once you have the data, it's just simple, so simple.
There's a bit of a danger there because a lot of correlations, if you're looking for correlation signals, to reconstruct some of the relationships between genes, let's say, a lot of the signal, pretty much most of the signal you get is from actually population heterogeneity. So in fact, it's more parsimonious to represent as different subpopulations versus, you know, connected, correlated genes. But if you control for that, then presumably, I mean, it can be worse than bulk estimation. And there are some papers that try to do it.
All right, so thank you very much for the introduction. We now know a bit better how this works, and we see it's very useful, so thank you again.